So our topic was on agricultural microbiology, specifically plant microbe interactions.
Hello, my name is Deval Coleman.
I'm Kiara Hodo.
I'm Zara Rich.
And I'm Leah Wright. I'll go to our next slide. So today for discussion, we'll be talking about how plant microbe communication enhances auxin biosynthesis by a root-associated bacterium known as Bacillus amyloliquefaciens, or SQR9. So just before we go into our presentation, you should know that auxin is a class of plant hormones or plant growth regulator, and it basically allows for many growth functions and behavioral processes in plant life cycles, and it's derived from tryptophan. And auxin biosynthesis reveals the sites of auxin production in plants, and it allows us to define auxin sources and where they're located. So to begin with our introduction, there are many root-associated bacteria that can increase crop yield, and they provide advantages over chemical fertilizers because chemical fertilizers can damage the ecosystems due to runoffs. So in the past, many researchers wanted to see the different interactions between bacteria and plants. So through the studies that they conducted, it was shown that indole 3-acetate acid, or IAA, allowed for plant growth and stimulation of roots. In this study, they wanted to demonstrate that plant microbe communication can contribute to auxin synthesis by utilizing Bacillus amyloliquefaciens, or SQR9, and basically promote plant growth because previous studies also showed that this may be linked to plant growth. They believe that this was able to be done basically because SQR9 is able to synthesize more IAA in the rhizosphere, which in turn should increase plant production. So we can go to our next slide. So just a continuation on the auxin synthesis. It is mainly synthesized from the amino acid tryptophan, and this is a two-step auxin biosynthesis pathway and is highly conserved throughout the plant kingdom and is essential for almost all of the major developmental processes. So directed in the picture below, it basically shows the stress factors and benefits just to give you guys a visual, and the benefits of each factor has an outcome as well. So the overall purpose of this study is to demonstrate that plant microbe communication can contribute to auxin synthesis by the root-associated strain by using Bacillus amyloliquefaciens, or SQR9, to promote plant growth. The hypothesis is that Bacillus amyloliquefaciens, SQR9, can be used to promote plant growth, stimulate the roots, and promote auxin synthesis.
Okay, so now we're going to get into our methods and materials for this experiment. So the first step in this experiment was to grow the bacteria and the plant that they were using. And one of the bacteria, of course, was the Bacillus amyloliquefaciens, SQR9. I'm just going to call it SQR9 for now. And all these bacteria were grown under the same conditions, and also a mutant strand of SQR9 was grown, and it's going to be denoted by delta YHCX. And then the plant they used was a cucumber plant, and they grew them under these specific conditions. And they're going to be inoculating as well as collecting exudates throughout this experiment. So the exudations that they collected were going to be extracted for their RNA. So 2 days after inoculation with SQR9, roots from the exudated roots were collected and cut and flash frozen, and then they were grounded into a powder to extract the RNA. After the RNA was collected, they did their process of evaluation, which can be read below. And before that, they also did inoculation for the, or exudation for the non-inoculated roots as well. So basically, they're being very thorough in this experiment with their observations of the RNA extracted. And then after that, they quantify the tryptophan in the roots that were exudated, and they use two different processes for this. They use the HPLC analysis, which is a powerful analytical technique used for the separation and analysis of compounds in a solution. And they use the GCMS, which stands for gas chromatography with mass spectrometry. And it's an analytic method that combines features of gas chromatography and mass spectrometry to identify different substances within a test sample. And then after that, they perform the plant growth promotion assay. And below, they did 6 different treatments to basically test all spectrums so they can monitor the plant growth. And they used a control treatment. They added only tryptophan to one. They added a 10 to the 5th CFU concentration, and so on. And here is just showing you basically each of the plants that were under different concentrations. And the final steps that they used were for the remainder of the experiment, a culture experiment was done. Excuse me. A culture experiment was also done to evaluate the IAA production from the bacterium, the SQR9, under different conditions. And then finally, the RNA was extracted from the SQR9 that colonized on the cucumber roots. And reverse transcription PCR was performed for the RNA that was extracted from both the cucumber plants and the SQR9.
So now we're going to discuss the results of this experiment. Next, you can go to the next. Okay. So first here I have the qualitative results of our experiment, and qualitative is just kind of giving a visual of what happened over the experiment. So in this visual, we have a plant that was used in the experiment, the cucumber plant. And on the left side of the plant is where it was inoculated by SQR9. And there are also roots who have no inoculation, and they are used as a control group. And then on the right side, there's three separate experiments that happen at the same time in this experiment. So there's 3 different areas of focus. One is for the excavation of the roots. One is for the extraction of the SQR9 RNA. And then another is for the cucumber RNA extraction. So here is the quantitative results, essentially describing or showing the tryptophan detection and root secretions. So the top graph is showing that the SQR9 peak is the highest for the tryptophan detection and root secretions in picture A. And then in graph B, they are showing the quantification of A. So that's just a different way of showing what's the information in graph A. And then graph C shows the gas chromatography quantification. So for A, they use liquid chromatography, and the roots were inoculated with SQR9. The Bacillus amyloleac contain significantly more tryptophan, and C shows that as well. Okay, and in this the next 2 graphs we see, the first one is showing how much IAA was produced. Production of this phytohormone leads to greater root surface area. So IAA production, and root secretions and verification of effect of YHCX mutant on root surface area. So that's the mutant that was mentioned earlier, the mutation of SQR9. So if we're looking at the first graph, we see that the tryptophan IA production is the highest. And then at the second graph, the root surface without inoculation are low, while the wild type with tryptophan is high, and then the mutant basically has no effect. I believe this is my last slide for results. So at the top, you'll see in picture A, another qualitative visual of how the plants grew, and you'll see in the second to last beaker, the plant has grown the most, and that has the most tryptophan involved. So A is showing the plant growth. Graph B shows the dry weight of the plants, and you will see that with the tryptophan, the dry weight is higher. In picture C is showing the root surface square, so the same as the root area essentially. And you will see that once again with tryptophan is the highest. And in D is the expression of genes related to IAA synthesis by SQR9 roots previously inoculated with SQR9. And the YHCX, which is the mutant of SQR9, has the highest result, or the highest gene expression.
And I would just like to add that inoculation is immunization, and then exudates are basically what the organism excretes, and that's what they're collecting from all these plants.
Okay, so in conclusion, previous studies have indicated that tryptophan-dependent N-nucleocetic acid, which was referred to as the IAA, its synthesis is a major mechanism by which Bacillus amyloliquefaciens, SQR9, promotes plant growth by producing more tryptophan. Clinical findings demonstrated that the colonization of cucumber roots by Bacillus amyloliquefaciens stimulated the transcription of the root tryptophan transporter gene, thereby increasing tryptophan secretion. SQR9 uses the tryptophan as a substrate for NO3 acetic acid synthesis, which benefits plant growth. Therefore, the collective results support the promotion of plant growth in the rhizosphere was affected by the SQR9 root exudation. Factors regulating the expression are poorly understood. Pretty much root secreted tryptophan is known to be the ability of to be important of the ability of many beneficial factors when it comes to the promotion of plant growth. Factors regulating the expression of the beneficial plant root traits are poorly understood because, just repeated that part, I'm sorry. As reported, the tryptophan in cucumber root exudates is not sufficient enough to detect the NO3 acetic acid production by the bacteria. However, cucumber roots does increase tryptophan secretion in response to the Bacillus amyloliquefaciens colonization, as well as increasing endo-3 acetic acid production and illustrating a bidirectional communication process between the rhizosphere bacterium and the plant. Alongside with this data, it showed an increased rate in plant growth by the SQR9, and these findings suggested that plant microcommunication could be an important component in plant growth promotion. So in this study, the beneficial bacterium from the tomato rhizosphere, which is a Pseudomonas brassicaserium J12, it failed to induce the secretion of tryptophan from cucumber root exudates. Similarly, the beneficial bacterium Pseudomonas fluorescens WCS365 from the potato rhizosphere did not increase the growth of the cucumber as well. And it secreted a low amount of tryptophan in the root exudates. Basically, the isolation of SQR9 from the cucumber rhizosphere may suggest that SQR9 crosstalk is a reflection of co-evolution of plant hosts and rhizobacterium. All right.